Doop, doop, doop. Get that in there before Rose does. Good morning, everybody. Almost Friday. Okay, so just got a couple of minutes before things start in full. If anyone has any questions about stuff in the meantime, feel free to fire away. I'm just making sure I'm refreshing my memory as to where we're at in. I think I remember where we ended up. Yeah. Oh, I didn't get myself a coffee. Oh, no wonder I'm feeling like I'm still asleep. I believe that we finished up, and feel free to correct me if I'm wrong. Hello, Tanya. I think we finished up, not quite ending the non-Mendelian inheritance PowerPoint. Which is this one here. I think that's right. That look familiar to you, Tanya?
I think we were left there. Well,
Good.
the slide before you talk about it, but that we didn't.
Okay, great. All right, so I might as well get cracking. I want to kind of tidy this up fairly quickly. I don't really want to spend too much time on it because it's fairly self-evident, to be honest. And also, I want to make the most of our linkage time, which is today and Tuesday. It's really weird how this semester's gone, like schedules all over the shop. Okay. All right. So essentially, the idea is that, and I've probably told you this already, but very, very few traits are controlled by a single gene. In humans, it's typically disease traits. I mean, in simpler organism, yes, right? So eye color and Drosophila is controlled by a single gene, right? It's the white gene. But in humans, given that we're far more complex genetically, there are few traits. Typically, they're disease-causing traits which are caused by a single gene. You know, sickle cell disease, cystic fibrosis, you know, Tay-Sachs. There's all kinds of different genetic diseases, right, which are caused by a single gene. There's a whole bunch that aren't as well. But even those single gene traits have multiple alleles, right? So I think cystic fibrosis has something on the order of 1,500 different alleles present in the human population, depending on which population depends on which one you're more likely to have if you do have cystic fibrosis, right? So even single gene traits aren't simple, but most traits in humans, really almost anything that you can think of in humans is a complex trait. You know, let alone obviously complex things such as intelligence, right, or academic ability, however defined that is, right? But even things which you might have been told are single gene traits like eye color and hair color are not single gene traits, right? Eye color is, last time I checked, 4 different genes, right? Which makes sense, right? Because if you look at enough individuals and stare into their eyes, you know, you'll see that some people have brown eyes, some people have really dark brown, some people have hazel, some people have flecks, some people are, you know, green, gray, blue. Clearly, that cannot be controlled by one gene, right? Because otherwise, you wouldn't have all those different phenotypes, right? And one of the classic traits, right? So, try and talk about, what are the classic symptoms or traits of characteristics, sorry, not to mix work, characteristics of these different things, right? So incomplete dominance, classic characteristic of that is an intermediate heterozygote phenotype and a 1:2:1 ratio in a monohybrid cross, right? So you should already know that. So you shouldn't be writing that down, Tanya. It should already be in your head. But for example, epistasis, right? You're always going to miss one of the four or more phenotype combinations in the F2s, right? So a classic characteristic of a complex trait is normal distribution, right? So if we'd done this with a bigger group and actually plotted this properly in lab at the beginning of the semester with height, you would see that the height of students in a class, particularly if you have a big enough group, right? Kind of 4 or 5 isn't really enough. You will see that height is largely a normal distribution, right? So I'm one of those over there because I'm somewhat taller than average. And there are some individuals that are over here, but most individuals will be somewhere in between, right? That's that classic bell-shaped curve of a normal distribution, right? So if you see a trait which has phenotypes that kind of cluster in that way, that's almost definitely telling you it's a complex trait, right? And so, go on.
But like, would height be I guess factored by the environmental factors? Because like I'm Mexican, and in Mexico usually even men have lower height, and
Uh-huh.
women we have low height. Here in the U.S., many like Anglos they're like they're tall. Even girls are tall. Would
Mm-hmm.
that be like a different kind of
Well, that's a gene
that
by
thing?
environment interaction, without doubt. So that was kind of the second part, right? Not only are complex traits typically polygenic, but also there's going to be a gene by environment interaction, right? So height is a great one to talk about. So, and particularly given the demographic in San Antonio, which is a mix of people from Central America, mix of people from Europe and elsewhere, right? So clearly the genetic pool from which your collection of alleles, whatever they may be, that control height, right, comes from is going to be different from mine, right? Mine's mostly Scottish and German, and who knows what else, right? We're a bunch of mutts in the UK, people from all over the place, right? And so the alleles that I have for whatever those genes are that control that trait are going to be drawn from that pool, right? And that pool will overlap with your pool to some extent, but there will be areas where you'll see alleles in Central America for different genes that you don't see in Europe, right? Simply because of their ancestral lineage, essentially, simply because they're separate populations, right, which didn't mix until uh, a few hundred years ago, right? Even if you go all the way back 500 years, that's not a lot of time, evolutionary speaking, right? So that's one aspect. But the other aspect is obviously an environment as well, right? And I don't know enough about Mexican history to really kind of speak accurately to how nutritional levels have changed over time. So I'm not going to do that because I don't want to make assumptions or be wrong, right? But the kind of the best way of thinking about it is, say you have individuals, and this is a gene by environment interaction, right? Say you have individuals from Yemen, right? Currently in the midst of I don't know what, civil war, humanitarian disaster, right? Individuals who are born and raised there during this time will be severely nutritionally limited, right? So their height, their growth will be stunted simply because they don't have the energy for all those rounds of cell division. Their bones won't grow as long because they don't have enough energy for their bone cells to divide. even whatever their genetic background, right? They will be below potential. Now, if you take instead of those newborns that are being now kind of growing up in that environment, you take those individuals, or you split twins, right? If you wanted to do a human experiment, and you put those individuals in, say, Northern Europe, right, or the US or wherever, right, somewhere where nutrition is not limited, right, you'll see that individual which has by and large the same genetic background, right, give or take, that individual will now grow much taller, right? So you can take individuals of the same genetic background and put them in different environments, and you'll see a different, either not necessarily a different phenotype or a different expression of that phenotype, right? You can also take individuals of different backgrounds and put them in the same environment, and you'll still see a difference, right? So it's kind of it's a complex interaction, right? And it's very, very hard to tease out. And this is kind of what I was talking about in lab a little bit yesterday in response to Jelly's question. It's very hard for that reason to tease out what causes certain traits in humans, because we have our genetic background is, I mean, like depends on how you look at it, right? We're almost entirely identical if you look at it from one perspective, and we're also very varied genetically if you look at it from another perspective, right? Depending on kind of like the scale at which you look at. Compared to chimpanzees, we're almost all identical to each other, right? Compared to each other, clearly we're not, right? We're clearly very different. But we also have a huge range of environments, right? So, you could look at, say, for example, people stayed in Mexico this whole time, people who emigrated, 40, 50 years ago, people who brought children 20 years ago and grew up here, people who came recently and whose children have grown up only in the US, right? What those environments and kind of genetic histories are going to be somewhat different, right? And also the environments are going to be hugely different, right? Whether people who are stayed in Mexico, what is their socioeconomic status, right? What's the quality of the schooling available there versus where they end up here? Is it better? Is it worse, right? Nutritional availability, types of nutrition, right? Diet is clearly somewhat different, right? Even if you bring a bunch of your familial recipes and things like that with you, obviously stuff is going to change, right? High fructose corn syrup, in everything here, maybe not so much in Mexico, right? And so,
There's a lot of hormones. I think one aspect of that is when you go over there and you see teenage girls, they're not as developed as
right.
teenage girls from here. When I came here, I guess I developed differently because I guess there's too much hormones in food here.
Yeah, I mean, that's quite possible too. But again, that's also very hard to figure out because is it just the hormones or is it the type of nutrition that caused different changes during that growth period? Right? Eating high energy foods, does that change how you produce your own hormones, let alone what hormones may be present in the environment? Right? So it's very, very hard to figure that out. Really, really hard. And so it's pretty much a given that whatever trait you have, unless it's a unless it's a basic pigmentation trait, I think, like things like hair color, eye color, skin color is a pretty poor one because obviously that reacts intrinsically to the environment, right? Sort of. I get just pink. I don't go brown. I just go pink. That's my kind of Northern European inheritance. But kind of stuff like hair color and eye color, those tend not to change much by environment. There's not much gene by environment interaction there. But a lot of other stuff, your immunological capability, right, depends on your environment, depends on what you're being challenged by in terms of, antigens, whether they're food-borne or pathogen-borne, depends on how well, how good your nutrition is, right? How well you're able to mount an immune defense, right? So it's all kind of, yeah, it's a lot of hand waving, right? Really, and it's very hard to actually scientifically tease out why you present a particular phenotype or expression of that phenotype for those. I'm not really going to talk about these examples. This one is actually kind of interesting in kind of a non-human perspective, right? And so this is actually a kind of interesting issue from the perspective of like monocultures, like Western agriculture, right? I'm not much of a botanist, but this is something that interests me. So typically in Western agriculture, like if you go into Nebraska, Oklahoma, they're monocultures, right? They're huge, huge expanses, literally as far as you can see, of exactly the same plant with exactly the same genotype, right? And so those plants are very highly, kind of tuned or bred or engineered in some cases to be high performing in kind of very rich environments, right? They have lots of nutrition, they have lots of fertilizer, weeds and other pathogens are aggressively suppressed, right? They're in the kind of like their most ideal environment possible, right? But if you try growing one of those genotypes, one of those strains, I guess, of wheat, let's say, or corn, in somewhere that's a little bit shittier, for example, if there is a drought or like, I think it was last year, there was some really bad floods in the Midwest, where those whole like millions of acres were inundated. That particular genotype will do very poorly, right? Now you can contrast that with, say, a kind of strain which is grown in more of a developing country, right, where they don't have the funds or the wherewithal to actually buy these super high performance strains, and they don't have the funds for fertilizer and pesticides and so on and so forth. Those strains will typically be what I'd say more robust, right? And it's a trade-off, right? So robustness versus performance, in a sense. And that's something that plant breeders obviously, tangle with all the time, because this other strain will be much more capable of coping with poor conditions, right? So if you're in an environment where the environment is not necessarily stable from year to year, or you're prone to floods or droughts or plagues of locusts or whatever, then you can trade off some of that performance in time when times are good to be able to still produce when times are bad, right? And so that's another way of thinking about this gene by environment interaction. Are you very, very specifically suited to one particular environmental set of circumstances and you fare poorly elsewhere, right? You're a specialist, essentially. Or are you a generalist that doesn't do as well in any particular environmental niche, but are able to succeed in more of them, right? And so that's in our hands. That's a specific decision that we make in terms of breeding plants. But in evolutionary terms, that's a trade-off, right? It's an evolutionary trade-off. You can do very, very well at succeeding in niche, right, and outcompete all your competitors. But if that niche disappears, you're up the creek. Whereas if you're a generalist, you may risk being outcompeted, right, and not succeed that way. Sorry. But if that environment does change, you're more likely to survive and continue, right? So that's kind of another way of thinking about this gene by environment interaction. Not just as in you as an individual, right? How do you react to the environment? But also how, as a species, you are suited to your environment as well. It's just another way of thinking about it. All right. Anybody got any questions? Obviously, there's lots of hand waving with this because it's very hard to talk specifics, right? Even with things that are, clearly different, it's still very, very hard to figure out exactly what's, you know, the cause, right? Are people, like you were saying, Tanya, in Mexico, where you're from, shorter simply because that's the genetic mix that they have from their ancestors and ancestors and ancestors?
Well,
Or is it
doctor,
because potentially the nutrition isn't as available or rich or whatever as it is here? You know, it's
would like a phenotype change, or like what you're born with the genes, or is that phenotype can express? And I asked because when I was little, my dad, he's 6'2", but he's Mexican, but he's 6'2". I don't know why. And my mom, she's really short. She's 5'1".
mhm.
So when I was growing up, they always thought that I was gonna be like my dad, because I was always like the highest from my school. But then I turned vegetarian, so I changed when I was a teenage, and I stopped growing and I stayed there. So would that be like something like the phenotype changed because of my like nutritional intake, or was that like already like done when I was born?
Absolutely cannot tell you. No idea. So that could be something that, and the reason why I can't tell you is because there's not another one of you that did not do that, right? And so really the only way to ask those questions is with twins, and then to have those twins through chance grow in different environments. And there's a few studies of that, you know, when they've been separated at birth because of, uh, being orphaned or divorced or whatever, right? Not them, their parents, obviously. Um, so that's really the only way that you can answer that question. In your case, there's no way of knowing because it could be that that was just people grow at different rates, different times in their development. The whole growth spurt thing can also go the other way too. So it may have just been that that was what you were going to do anyway. It may have been that, you weren't noshing down on those lovely beef growth hormones anymore, right? Could be that the nutrition was poorer, although if you eat well as a vegetarian, you can have just as good nutrition as if you eat meat. You just have to be more conscientious about it. So there's no way of, you can pick whichever answer you like, right? But there's no way of really knowing what one of those, whether that was the phenotype you're always going to have, or if it was some result of the environment changing, i.e., your nutrition. Couldn't tell you.
Okay, yeah, and it's like a control to see the
Yeah,
difference.
exactly. And that's why it's very hard to do these things in people because we don't have those controls, both biologically and also ethically as well. Okay, so I'm gonna, if anybody else have any questions, shout them out while I kind of get over to the next one. All right. Now we get into the weeds. All right. So hopefully, and it's kind of weird again. Um, get over it. Because of the change in the lab schedule and the change in the class schedule, things aren't lining up quite as well as they should do. So hopefully you have all, maybe, by now had some exposure to linkage, either in the lab or in the other sections. So I don't know how this is exactly being taught in the other lab sections. So hopefully you're also getting this in those lab sections too. Okay, so essentially this is about distance, physical distance. And really up until now, uh, our kind of view of inheritance, right, within, this course has been that, genes behave independently, right? So whatever alleles you get for one particular gene, doesn't have anything to do with the alleles you get for other genes. Now, clearly that's nonsense, right? Because from your own family histories, right, there are certain traits which kind of go down through generations, right? And often those traits are they kind of go together, right? One of the classic ones, particularly in my family, is allergies, right? So allergies, eczema, and asthma, all three of those things, even though they're not necessarily caused by the same genes, tend to travel together, right? And so somehow, we don't actually know, I don't know the genetic basis of those different traits. Those tend to kind of, if you get one, you get at least one of the other ones as well, right? And so the kind of common vernacular way of talking about this is that genes travel together, right? And so what allele you get for one gene, you'll almost certainly get a specific allele for the other gene at the same time, right? So they're kind of going down the inheritance kind of path together. They're not swimming in a separate stream on their own. And really, when you think about this, it kind of makes sense, right? So these aren't genes, they're actually linkage groups, which is something a little bit different. But we have 23 chromosomes. Yeah, 23. And we have approximately 20,000 genes, right? So we have obviously more than one gene per chromosome. We have ranges from, I think, 150 to about 1,500 genes per chromosome, give or take. And so clearly some of those genes are going to be near to each other, right? They're not going to be far apart. And so linkage is really about how close are those genes, right? So that's one aspect of linkage. The other one is just simple probability, mechanistic probability based on distance. And that probability is a probability of a recombination event, right, which always happens in meiosis in prophase 1. The probability of that recombination event occurring in any given patch of the chromosome. And so really, other than the telomeres and the centromeres, which are telomeres at the end, centromeres in the middle, recombination frequency is the same all the way across, right, a chromosome arm. So the probability, for example, of a crossover occurring up here is exactly the same as the probability of the crossover occurring here, or here, or here, or here, or here, or here, anywhere, right? So you have a given probability per kind of distance unit that in that round of meiosis, recombination will occur in that patch of DNA, right? And so this is the addition rule. So if you have probability, let's say, of 1 in 100, right? Actually, I'm going to do a little bit smaller. Recombination is going to occur in that patch. Now, if you look at a bigger patch that's 5 times that size, probability is going to be 5 100s. Look at a patch which is 10 times that size, the probability is 10 100s, or 1/10. 100 that size, probability is 100, right? Or 100 out of 100. So probably, like, actually, you can't see my hands. There's one over there, and there's one over there, right? Probability of recombination occurring between those 2 points is 100%. It will always occur somewhere. We don't know where, right? Could be here, could be here, could be here, could be here. Somewhere between those points, you will have a recombination event, right? So really, that's the key thing that you have to understand linkage. It is a simple matter of probability based on distance, whether or not recombination will occur between those 2 points. The closer the points are, the lower the probability recombination occur. The further points are apart, the higher the probability, right? That's the key kind of absolutely central point you have to understand. If you get that, everything, pardon me, everything else will fall together. Oh, is he gone? Our foster puppy is gone. Oh, I'm sorry, love. My wife is sad. Okay. But we'll get another one tomorrow, though. Yay. Okay. Back to the program. So essentially, if genes are on different chromosomes, you would expect to get equal probability of getting any gamete, any allele combination in the gametes, right? We're talking about two genes here. These genes are on separate chromosomes, right? So which way round, and these are heterozygous individuals for both genes.
I think we're still on the other lecture slides.
Are we? You're not
Yeah,
seeing linkage?
I'm seeing like the crop graph.
That's really weird. Let me stop share. I'm going to actually close that PowerPoint, and then you definitely can't see it. Get out of it. That's so weird. Usually whatever I see, you see. Better?
Yes.
Yeah, weird. Maybe I selected the PowerPoint rather than screen to share. Anyway, so thanks for that. I'll never known otherwise. You'd be wondering what I was talking about. These are heterozygotes for both genes. And essentially due to random alignment on the metaphase 1 plate, right, which way around those homologous chromosomes happen to align, you essentially get equal numbers of, dominant, 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 recessive, recessive, dominant, recessive, recessive alleles, right? That will give you a 1-to-1 to 1-to-1 ratio, right? So if you think back to dihybrid cross in Mendelian inheritance, the proportion, you get 4 different gametes from a heterozygote, from an F1 heterozygote, and they're present in equal proportion, 1-to-1 to 1-to-1. Similarly, for the other heterozygote, you get the same 4 different possible gametes, also in the same proportion, right? So this is independent assortment. This is those genes are assorting independently of each other, right? Now that obviously has an additional wrinkle if those genes are on the same chromosome, right? So now what we have is a recombination event. This always occurs every round of meiosis 1, right? This is what holds together the homologous chromosomes up until metaphase 1, right? Without it, they'll just get pulled apart and the whole caboodle wouldn't work. Also, this is another way, if you think about it, of fine shuffling those cards, right? So what alleles your offspring get obviously depends on what alleles your partner is contributing to that offspring. But whatever collection of alleles you have, you're going to be essentially providing a new combination to every single gamete that you produce, just by the independent assortment, that random alignment. However, even more finely, you're going to shuffle that by creating new chromosomes. We're not talking about new chromosomes as in like chromosome 24, 25. What we're going to do is we're going to take these parental chromosomes, right? This is recessive for both genes. This one's dominant for both genes. And through recombination, you're going to generate not just the parental chromosomes, right? We've got one here, the recessive, we've got the dominant down here, but you're going to generate two non-parental chromosomes, right? Chromosomes that weren't present in either of your parents. These are also called recombinant chromosomes because they're the result of recombination. And these will have a different collection of alleles, right? So now you have a chromosome which is recessive dominant, and another one, because it's always a mirror image, it has to be, is going to be dominant recessive, right? So these are chromosomes that were not present essentially at birth of you as an individual. They're new combinations. Now, if these two genes are so far apart, remember that kind of probability deal, right? If those two genes are so far apart that every meiosis, there will be a recombination event between them, right? And this is why they're kind of drawn at either end of the chromosome arm, right? Guaranteed, without any shadow of a doubt, there will be recombination occurring between these two. Then essentially, these genes are behaving independently. Because every round of meiosis, you'll get an AB, an A plus, a plus B, and a plus plus. Just the same as if you wanted to use different letters rather than A and plus, or instead we're using capital A and lowercase a. Here you get essentially plus plus, plus B, A plus, AA, AB rather, sorry. Right, same kind of nomenclature, right? Really, saying the same thing. But you'll get the same ratio. You'll get, for every round of meiosis, if, again, and that's an if, they're far enough that recombination will always occur, you'll always get one of these, for one of these, for one of these, for one of these. Literally, there will be 4 gametes, and each one will have a different chromosome every time. Different, not as in they're different numbers, obviously, I want to stress that, but that they have a different combination of alleles. We got a question. Ooh, question. If I can get it up. Ah, that is you, Tanya. You could just tell me it. Save me all this hassle of looking for the chat box. No, is the answer. So you'll never get recombination between the chromatids on the outside of that pair. Recombination only occurs between the non-sister chromatids that are next to each other physically, right? So you can't get that, essentially. You can only ever get that. So these ones on the outside, they're wiggling, these ones. Tanya. Those ones never recombine. It's only these ones, the ones next to each other. So that's why you will always get, parental chromosomes go through, whether or not recombination occurs. So if it does, you'll still get parental chromosomes. There'll just be 2 of them. And you'll get 2 non-parental or recombinant chromosomes, these ones in the middle here. Right. And if it occurs all the time, these will be in a 1:1:1:1 ratio. Good question. However, not all genes are so far apart on the same chromosome that recombination will always occur between them. Some of them are closer than that. And drink of water for a second. So if they are close enough together that recombination does not always occur, you'll get one of 2 possible outcomes, from that round, from any given round of meiosis. Obviously, what you get from one round of meiosis is what you get from one round of meiosis. But meiosis doesn't happen just once. It happens every time you generate gametes, right? So if it's, let's say, it's not this far apart, it's kind of this far apart, right? So there's a probability that recombination will not occur. However, there's also a possibility that it will occur, right? Again, it's just probability based on distance. Pardon me. So if probability occur, that if recombination does occur, you'll get exactly what we talked about. You'll get a mix of parental, what you saw over here, and recombinant chromosomes. Yeah. However, meiosis happens again, there's the probability that recombination will not occur between those two genes. It will occur somewhere, right? It's not that it just doesn't occur at all. It's just if you have this distance, you could have recombination occurring over here. But you're never going to see that because this is what you're looking at, right? So recombination will occur somewhere. It's just, it might not occur between the two things you're looking at. And if that doesn't occur, you'll get only parental chromosomes. Only non-recombinant chromosomes will be produced in that round of meiosis. Right? But now we're going to look at all rounds of meiosis, right? Because you're looking at offspring, right? Maybe, humans are really not very good at this because we only have, 2 or 3 or 4 typically. But if you look at, corn plant, let's say, or some kind of animal that produce lots of offsprings, like mice or something like that, or rabbits, kind of always think of Zootopia whenever I say that. Anyway, when you look at the whole of the offspring, my dog's come down, then you'll see the result of those rounds of meiosis where recombination didn't occur, right? And so essentially, all of that basically means that if recombination doesn't occur, you'll get more parental non-recombinant chromosomes going into gametes. And so in the offspring, you will see more allele combinations that are like what were originally in the parent. You'll see fewer of the allele combinations that arise from recombination that were not present in that parent, right? And so that really is the key characteristic of linkage. You will see more parental, there's an excess of parental combinations, allele combinations, or phenotype combinations, whichever, that's what you see. There will be an excess of parental gametes, chromosomes, phenotype combinations, whatever you want to call it, then there will be recombinant. So if you go back to the whole dihybrid cross deal, right? And you have, actually we've got, rather than just telling you about it, for example, right? We have a dihybrid cross between these two parents. One can assume that this is the dominant, right? Because that's what you see in the F1s, recessive. Cross these together, get heterozygotes. What ratio should you get of different phenotype combinations in the offspring? It's a dihybrid cross.
9:3:3:1.
Right, exactly. Is this a 9:3:3:1 ratio? No, not even close, not by a long shot, right? Instead, what you see is fewer of these So in a classic dihybrid cross, like we started learning, these genes, the genes encoding these traits would be on different chromosomes, right? And so you get a 9:3, 3:1 ratio. However, it's pretty clear that these genes are on the same chromosome and they're pretty close together because these are not assorting independently. We are not getting a random distribution of alleles in our gametes because we're not getting the expected ratio of offspring in the F2 generation. Right? We should be getting a 9:3, 3:1. What we're getting is, shoot, I don't even know what that is. 10:1:1:3, right? And so this is the classic characteristic of linkage. You're not missing a phenotype combination, right? That would be epistasis, right? You are seeing an excess of parentals. That's these ones, right? That's what you see up here. And this one over here, again, that's what you see in one of the original parents, hence parental. You're seeing far fewer of these different combinations, right? Purple and round and red and long, which you do not see here. So normally in a classic independent assortment dihybrid cross type deal, these traits would be on different chromosomes, and so you just get different combinations, essentially, of alleles from your F1, right? That's why you get these different combinations of phenotypes. However, they are still caused by different genes, but now they're not behaving independently, right? So if you get the allele for purple flowers along if you get the allele for purple flowers, you're much, much more likely to get the allele for long pollen, right? So these alleles are traveling all the way through this cross together into this parent. If you have an allele for round pollen, you're almost certainly going to have an allele for red flowers, right? And so you can see that combination, red flowers, round pollen, even though it disappears here because it's recessive, it will still pop out together more frequently down here. Those alleles are traveling together, right? And so this disruption of your expected, or essentially disruption of independent assortment, which then leads to this odd looking ratio, that is because of linkage. And it's always, you'll never see an excess of recombinants, right? Again, because if you go back up here, you always get parentals, even if a recombination occurs, you always get, dog's crunching a bone right next to me. You always get non-recombinant parental chromosomes coming through, right? So you can never have more recombinants than non-recombinants and parentals. You can only have less. And when you have less, it's because these two genes, flower color and pollen shape, are linked. Not completely, because you still do see recombination occur between them, but these are fairly closely linked. All right, get people to catch up and think. Any questions?
Yes, so what would be because I'm getting confused like with the epistasis, because in the epistasis you also get like a funky kind of ratio. So to differentiate with the linkage, you're gonna have an excess of the parental, and the on the epistasis that's not gonna happen. Like you won't see
Oh
too
no,
much of
what happens in epistasis is you don't see one of those four combinations. So dominant epistasis, you wouldn't see, oh, which one would it be? You wouldn't see red flowers and long pollen. Is that right? No, sorry, the other way around. You wouldn't see purple flowers and round pollen. Just got to think through. Never done it for that one. In recessive epistasis, you wouldn't see, essentially, you wouldn't see 4 different phenotypes. You'd only see 2 different phenotypes. It'd either be purple flowers or it'd be red flowers, right? And so you wouldn't have 4 possible combinations in the F2. You'd only have 2. It'd either be purple or be red. And so that's the way to differentiate between linkage and epistasis. Linkage you're still seeing, or at least unless they're like absolutely completely linked, in which case it would be pretty difficult to distinguish between the two. Yeah, actually, we'll get to that in a second. You'll always see the 4 different phenotype combinations, both in the parents and in the F2 generation, but they won't have the expected ratio in the F2s. In epistasis, you'll have maybe 4 different phenotypes in the F2, maybe it'll be 3, maybe it'll be 2. Sorry, in the P generation, in the parents. And in the F2 generation, you will see less than 4 different combinations. And there are all kinds of different ones as well. Oh, by the way, I think it was this group we talked about rhesus factor. Just remembered. I spent like 10 minutes trying to understand that this morning. It's another antigen on the red surface of the red blood cells, but the genetics are absolutely crazy complicated. So I'm not going to go there, basically. Actually, I just want to show you just because it's funny. Hang on, I don't want that. Here we go. Okay, so these are all the different phenotypes and genotypes you can get for rhesus factor. So I think I counted them at 19, I think, in total, which gives you either positive or negative, right? So I am never going to teach that in a class, not even once, not even in like advanced molecular genetics. Just no, I'm not going to do that. Okay. Any other questions? Just thought I'd share that because that was funny. I looked at that, I was like, oh my gosh. Like, and there's like 2 competing kind of, well, there's actually 3 different competing arguments about how to do it. Anyway, so one kind of heads up that I want to give you too is that you have to work out what's going on from scratch. You can't make any assumptions, right? So we drew this. Actually, it's kind of annoying how we go back from different kind of terminology or notations, right? We drew this as this chromosome having both recessive and this chromosome having both dominant alleles. You can't assume that that's going to be the case, right? When we get into, where are we at? Down here, look at that, feast your eyes, right? You can't assume that the gene order is going to be in a particular way, right? It's going to be ABC. It might be ACB or BAC or whatever, right? You cannot assume either gene order or linkage phase. Back up here, right? Before you go into the problem. You can only work out linkage phase and gene order by looking at the results of recombination in the offspring. Okay, so in lab we talked about essentially the flies that we're working with have this linkage phase. This is called cis linkage. And so if you say that they're in cis linkage, that tells you that the 2, obviously they're heterozygotes because otherwise you'd never see recombination. But if they are heterozygous, then both dominant alleles are on one chromosome, both recessive alleles are on the other chromosome. That's what cis linkage tells you. Translinkage means we have a recessive and a dominant on one chromosome, and then the opposite on the other chromosome. Dogs. You cannot assume that, right? And I want to be like totally upfront about that. You will get problems in the exam, guaranteed on this, right? We're spending 2 whole lectures, like 2.5 hours talking about linkage. You can put any amount of money on this that you'll get questions on linkage in your exam. But you cannot assume that I'm giving you the correct gene order or that I'm giving you the right linkage phase. You can't assume anything. You have to work this out from the numbers. Okay? That's not me being tricky. That's simply because you cannot tell without actually looking at the numbers. Unless you knew beforehand, in which case, like, well, you know, whatever, right? If you already know what the gene order is, then you're not going to be doing 3-point cross to figure out the gene order. It's kind of a waste of your time, right? So remember that. And I'll give you some problems on Tuesday. We'll actually go through particularly the 3-point cross, which tends to trip people up. I'll give you some problems where that's the case. You're not going to be going into this blind. We're going to have plenty of practice. Okay, so so essentially, you know, this is kind of interesting from an inheritance perspective, linkage, right? Because it means that you'll get certain combinations of phenotypes for specific traits. If those genes are linked and you get again, asthma is a great example. If you get asthma, then you're almost certainly going to have allergies like hay fever, let's say, or eczema, right? At least 2 of those go together. Maybe if you're particularly unlucky, you'll get all 3, right? So it's interesting from that perspective, but it's also an extraordinarily useful tool, right? So we can actually work out how far apart genes are on a chromosome based on how many recombinants we see. We can figure out gene order on chromosomes. We'll get to that maybe in a little bit, right? And so essentially, so this is flies again, right? Whew, I had enough of those in lab, right? But we're looking again at a couple of X-linked traits. And actually this morning I was working out on my really tatty notepad a 3-point cross with these traits. It's actually kind of neat. That's why I was looking on Flybase. Right, so we have white-eyed females and red-eyed miniature wings. Okay, quick test. What linkage phase? It's going to be all me telling you stuff. Come on, tell me stuff.
Like for the females, wouldn't that be the cis or no?
No. That would be, so what you're looking at, so this is essentially, if you remember from lab, what's above the line is one chromosome. What's below the line is another chromosome, right? So going back, get off here. Essentially, this is, so this is actually, this is even trickier, right? Ha, us geneticists, we're all a bunch of tricksters, right? This is a heterozygote, right? So it will have one copy of each allele for each gene, right? This is a double homozygote because it is homozygous recessive for W, so it has white eyes, and it's homozygous dominant for M, so it has normal size wings. And so it has a dominant allele and a recessive allele. So this would be translinkage. And you can tell even more
So,
clearly if you look at the F1s where we have a heterozygote. So if you look at
so
these,
cis, it means they're gonna be like homogenetic, and trans are gonna be hetero.
no, it's not about their genotype. It's about which chromosome, what alleles you find on each chromosome. So if you find different alleles, like a dominant for one gene and recessive for another gene, that's in trans. If you find one of each, right? So for one gene it's dominant, the other gene it's recessive. Hang on, what did I say? Shit, I'm confusing myself. Sorry, let's go back a little bit. If you find one of each, it's in trans, right? So dominant for one gene, recessive for another gene, that's in trans. If you find both dominant or both recessive for the different genes, right? So again, we're just talking about one chromosome. We're not talking about genotype, which is, essentially what do you have for both chromosomes? We're talking about just one chromosome, like the haplotype, essentially. If it's dominant for one chromosome, it's cis. If it's recessive, it's cis. If it's dominant recessive or recessive dominant, it's trans. And so the bit that will trip you up here is that we have homozygous recessive and homozygous dominant. Now, if this were in cis, this individual, these females, would be white-eyed and miniature winged, because there would be homozygous recessive for white eyes and for wing size. So it would be WM over WM. That is cis linkage. And if you have the situation where the parents are homozygous, then you look at the heterozygous offspring. That will also tell you what linkage phase. So if this individual was homozygous recessive for both, and this individual was dominant for both, you'd still get a heterozygote, right? But what you'd see would be WM over, or plus plus over WM, right? If they're in cis, but you don't, it's W plus over plus M. Those are in trans. But you're right, it is easier to see in an individual that is heterozygous. Because then it's like, are they similar that way, or are they kind of like, similar that way? Does that make sense? Yeah. This one's actually a little bit tricky. I actually have to go think about that for a second. All right, so just as in lab, you need a heterozygote to be able to see recombination, right? Because if you have recombination between these chromosomes, what are you going to get? Another W plus chromosome, right? You're not going to be able to tell whether or not recombination occurred, right? So you need a heterozygote, and we're going to cross that back to a homozygous, or a recessive individual. And so we will see the effect of recombination between these two loci in the offspring. Now, again, put your hand up like in front of the screen. How would I do that? Haha, there you go. Can't see it now, can you? Right. What? Sorry. It's like the power of the professor. What ratio would you expect, right? So this is a backcross, right? So this isn't a classic dihybrid cross, we're not crossing these individuals with themselves, partly because we can't, because they're Drosophila, right? So these aren't going to cross with themselves. They're going to cross with a recessive male. So whether the offspring of male or female, you'll only see the expression of the alleles, essentially, from the maternal chromosome, right? And so the parental maternal chromosomes will be W plus or plus M, right? So those individuals will be either white normal wings or red eyes small wings, like these parents here. The recombinant should be plus plus, so red-eyed normal, and WM, white-eyed miniature. Does that make sense? So you're going to have 4 different phenotype combinations. What ratio would you expect if those genes were not linked and recombination was always occurring between them?
The 9331, right? If they're not linked?
No, because this is not a dihybrid cross. This is a back cross, right? So again, this individual here is recessive, and so it doesn't contribute really anything to the phenotypes in the offspring, because these are recessive alleles. So it doesn't matter if the individual had a W or an M, really. All you're going to see is whether or not it had a W or an M from the mother. So it's not a 9:3:3:1 ratio. You need to go back to here. It's going to be a 1:1:1:1 ratio, right? Because all you're going to be seeing is the different combinations of the 2 alleles that come from the mother. Now, again, what ratio would you expect if there is no linkage?
The 1:1:1:1.
Correct. It's always hard to remember how many ones you said, right? But yes, equal ratio, right? So if we look at 1,000 flies, you'd expect roughly, right, 250 to 250 to 250 to 250. Is that what you get? No. Right, so there's 644, so that should be what, 160? Yeah, give or take, of each of these 4 different ones. You do not see that, right? So first you look for where are the parental combinations. It's white eyes, normal wings, tick. Red eyes, miniature wings, tick. So if linkage is occurring, you would expect those 2 parental phenotypes to be present in excess. What do you get? They're present in excess. So linkage is occurring. And these ones down here, these bottom 2, you know that these are recombinant for 2 reasons, right? One, they're present at a lower rate. That's one way. That's not the most accurate, because if they're not linked, they can still be recombinant. They're just not going to see linkage. The other is that you have combinations of phenotypes that you do not see in the parents, right? So we have red eyes, normal wings, no red eyes, normal wings up here, and white eyes, miniature wings, no white eyes, miniature wings up here. So A, these are a different combination. B, they're present at a lower percentage or rate. So these are the recombinants. And the way you figure out recombination frequency, let me just tap this up. Oh, I'm sorry, Pedro, I didn't see that. Yes, you're 100% correct. I didn't get like a flashing notice that someone had put something in the chat. Yeah, so you get the chocolate treat, not Tanya. Okay, well, that's a virtual chocolate treat. Doesn't taste as nice as the real one. Right, so recombination frequency. As you probably know from being in lab, those of you who are in my lab, I don't have abnormally large fingers or hands. It's just because my camera's down there. So essentially that's number of recombinants. Move this over. Let's do this properly. Over total offspring times 100. Oh, actually, no, that's not. Now that's just recombination frequency. And so in our case, this is, what's that? 216, right? So that's both of these, right? You have to add both of the recombinants together because you can only get one by having the other, right? So you have to put these two together. And that's divided by the total, which is 644. And that is 0.335. Now, that's typically expressed as a percentage, right? In which case, or map units. And that's essentially the same, just times by 100. Right, so that would be 216 divided by 644 times 100 equals 33.5%. Or we actually have a unit, which is centimorgans, is 33.5 centimorgans. And so for 2-point crosses with just 2 genes, it's pretty straightforward. You find the 2 groups that have the lowest numbers, add those together, and then divide that by the total number of offspring present. Times it by 100, and you'll have recombination frequency as a percent, or in map units as centimorgans, or cm. Okay, here's another one. Here we've got the same parent. We've got a different, same female parent, sorry, different male parent because we're looking at different gene. This is now diminutive males, or DM, right? It's not small wings, it's small males, normal wings. Do exactly the same. Again, this actually tells you now. I was going to ask you that question, but that isn't going to work. Right, again, these are still in trans. You can tell most easily from the heterozygote, but also you can look at the parents. You can see that too. Now, are these 2 genes closer together or further apart than miniature wings and white are, right? So we still have white here, but we have a different gene instead. Are these two genes closer together or further apart? Is DM closer to white or is miniature closer to white? So what would you expect if two genes were closer together? What would happen to the probability of recombination between them? Would it increase as those genes get closer together? You're going to get more recombination? Or would it decrease as they get closer together? It would decrease, right? So if the probability of recombination decreases the closer together, what would you expect to see in terms of recombinants? Would you expect to see fewer of them as they get closer together or more of them?
Less.
Fewer, right? Much fewer, right? And so compare these numbers, 500 odd, to something in the tens, to these numbers. Which genes are closer together? White and miniature? Or white and diminutive males?
That one, the white and diminutive.
Right. So you can tell that because you get far, far fewer recombinants between these two genes. And so that's telling you that the probability of recombination occurring is much lower. We know that the probability of recombination is random and determined by distance. And so the distance must be smaller. And when you work that out, and we're just going to finish this up before ending for the day. When you work that out, we divide, what's that, 31 over 1,000. So again, if you look at what I posted in chat, recombination frequency, total number of recombinants, which are these ones down here, divided by the total, which is 1,000, times by 100, gives you the map units. So that's 3.1%. And that essentially is 3.1 centimorgans. Now compare that with what I wrote in the chat. This is 33.5 centimorgans. So generally speaking, these two genes are 10 times further apart. It's not a precise linear relationship, but it's good enough for our purposes. These two genes are 10 times further apart than these two genes. Right? So that's what I really want to get across. Probability of recombination in any point on the chromosome is the same. So the further apart two things are on the chromosome, the more likely recombination is going to occur because it's just based on distance. So recombination could occur here, 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 anywhere, right? Along that chromosome arm. So the further apart two points are, the more likely recombination will occur between them. And so we can use that when it doesn't always occur. It's telling us that they're somewhat close together than that 100% probability, right? And depending on how many times we see recombination, which is essentially recombinance, we can tell whether that's like that, like it is for white eyes and diminutive males, or kind of about like that, which is for white eyes and miniature wings. So now we know, relatively speaking, white is somewhere And those other two genes must be around there somewhere at different distances, right? What we'll do on Monday is try and figure out how we can order those. Is Y in between the two? Or is DM in between the two? Right? I'll show you how to do that on Tuesday. So I would strongly suggest, strongly, reading through the rest of the PowerPoint and maybe listening to me drone on about it in the podcast or whatever, and get a heads up on that. You have to make sure at the very least you understand what we're doing here, and ideally get a little bit of a heads up on what we're going to be doing here. Because as you can see, this gets much more complicated. Okay. All right, I went a few over. Sorry about that. But I thought that was worth it. Okay, so have a great weekend, everybody. Study hard. Don't have fun. You know, the usual. And I'll see you on Tuesday.